Hello everybody. Today I will be demonstrating an experiment which is very simple and which is going to be very useful to all of us. So, the experiment is going to be how to develop a bacterial culture. So, I am going to explain you all how to prepare a bacterial culture. So, what is a bacteria? So, bacteria are single celled organisms and they multiply in millions. For example, once they multiply, they can be present in any environment that is suitable for them, maybe even in our body, in different parts of our body, or maybe in some plants and even animals. So, once bacteria that are present inside our body, how do you recognize whether they are actually required for our body or are they going to be harmful to us? So, we know there are certain bacteria which are really helping us in everyday life, but certain bacteria when present inside our body and outside our body, they are going to be really harmful to us. So, then how do we know which bacteria is present or which kind of bacteria are multiplying in your body or outside your body? Don't you think you should know about it? Yes, definitely. Until unless we do not find out which is the bacteria that is present in our body and multiplying and can be infectious, which leads to diseases. So, we need to identify them. So, what is the methodology that we can use to identify which kind of bacteria is present in our body or anywhere where it is multiplying sufficiently? Once we know which bacteria is present, then we have a way to find out what is the treatment that can be done to cure the disease that we are carrying. So, then to find out which bacteria is present, what we can do is we can proceed for a bacterial culture, which is generally carried out in many of the clinics, as some of you all will be aware of it, like when some of your friends have been infected, then they ask for you all to take a blood test or submit your urine sample or some sample which will be cultured in the clinics, in the labs, and then they give you the result. So, in the same way today I am going to say you how to prepare a bacterial culture. So, I have a petri dish here, and in this petri dish I have prepared an agar nutrient solution. So, I have taken around 1.4 gram of agar and dissolved it in water and have prepared this nutrient solution. So, I have poured them into the petri dish. So, this would be the medium in which the bacteria is going to grow. Since bacteria is present all around us, what I can do is that I can just take a sample by just rubbing my hand over this particular surface and then get some sample for the bacterial culture. So, I am just wiping my palm, assuming that there were some bacteria present over this. So, now my palm is sufficiently dirty and now I am going to place this onto the nutrient medium. So, this and one more thing that all of us must remember is that all these bacterial culture has to be done in a sterilized chamber. Since it is for the sake of demonstration, I am just doing it here. So, I have imprinted my palm on this nutrient medium. So, I have labeled it and then now I am going to incubate it so that the bacteria multiply and then later it can be used up for further studies. So, I have kept them inside the incubator and then let us see if the bacteria has multiplied or not. It is 2 days now. Let us take our sample from the incubator and check whether there was a bacterial growth or not. So this is a sample. So there is the bacterial growth that you can see. So these are my fingers which I had kept, the 5 fingers, and you can see the bacterial culture or the colony that is present here. So now once a bacterial culture is developed, it will be used further in the labs, in the clinics, to test which kind of bacteria was present on this particular surface, or in case if a person has infection or disease, then it will be studied for which infection or which disease the bacteria has caused, and then the particular treatment will be rolled out. I hope you enjoyed it. See you all once again. If you all have any questions, please let me know in the comment section.
We are making these videos so that science learning can be fun and enjoyable. If you like our videos, please subscribe and don't forget to press on the bell icon so that you can keep getting the updates about our latest videos and experiments.